Yeah, I'm
Good afternoon. Welcome. Thank you for being here today. We're broadcasting live from Park City, Utah, where I am, and Peter's in South Africa. And where exactly?
Cape Town.
Cape Town. And where
Most
are you?
beautiful
Rene?
city on earth.
Yeah. Where are you, Rene?
Columbus, Ohio.
Oh, great. So thank you all for being here today. We have about an hour to spend together. Rene and I met some time ago, right? It was way pre-COVID.
Yes, we did.
Yeah. So we can't hear or see you, but we know you're there. And so we encourage you to put your questions in the chat box in the Q&A. And we have a bunch of folks coming on today. So just post them as they come to mind so we can get to them as soon as possible. We're going to be recording this and posting it to the Family Office Insights YouTube channel and perhaps somewhere on Neurxstem's site, which would be better if we could do that, just to drive people to your area there. And then we're going to be also connecting you directly with Rene, so you can, if you're interested, you can chat further about the funding requirements. And then we'll also be doing another webinar to bring in more of the Family Office Insights community after having seen this today. And also keep in mind, if there's anybody that you think will benefit from hearing what you hear today or connecting with Neurxstem, please feel free to do that. We'll make sure everybody gets connected. So with that, please, Rene, take it away.
Good afternoon, everybody. Thank you for coming. And of course, Arthur, we have met before. This is the second time. Thank you for hosting us on Family Office Insights. It's really exciting to be here. We have a very exciting presentation because we are at an inflection point in brain medicine, and I hope you will realize that after my presentation. What I would like to do today is to introduce our incoming Chief Operation Officer, Peter Cruz. He's a scholar, Rhodes Scholar. He's a fellow of the Royal Society of Medicine in London, and he's on our team. And Peter is going to give you the high-level summary of what is in the webinar, and then I'll fill in the details. Peter, it's all yours.
Right, thanks very much. And again, thank you, Arthur, for the invitation, and Rene. We were just discussing how I met Rene at an investor conference in London a few years ago before COVID, when I was acting on behalf of a family office from the Middle East. But my job at the time in Oxford was, where I was based, was working with the medical Rhodes Scholar startups in Oxford and helping them get to market. And so my job in this team is to take the discoveries and the technology that Rene has worked on and turn it into a viable, marketable, and investable product. Now, the story you're going to hear today is summarized on this slide. We're delighted to present an unprecedented investment opportunity that holds the potential to really redefine the landscape for neuroscience, human neuroscience. I'm a medical doctor by training. I have a PhD, an MBA, and 2 board certifications as an anatomical pathologist. So I'm a tissue pathologist. And you're going to hear a little bit about what Rene has done with brain tissue. And he'll explain to you this amazing discovery of the Neurxstem brain model, which is essentially a bit like the old days of microscopy culture sensitivity, when we used to take a swab or a piece of somebody's urine or blood and grow it in a test tube to see what it was doing, which antibiotics worked and which didn't. And Rene has, in a nutshell, done that with human brain. He takes a skin cell and he talks to it through very clever chemical means. He grows this piece of stem cell into a blob of tissue, which is a very accurate model of the human brain. So it comes from a patient with the disease, in this case, Alzheimer's disease, and then he interrogates the tissue by asking which genes are switched on, which are not working, and how do they compare with the brains either of living or post-mortem patients who've had similar diseases. So it's a completely novel approach. And this is why I sort of gave up my day job to come and join Rene, because I think this is potentially an inflection point which could change the history of human medicine if we can back him and get him to market. Now, Neurxstem is a visionary biotechnology company, and it's looking for partners to embark on a journey of taking the discoveries that you're about to hear to market. In other words, to do the clinical trials that are needed to prove safety and efficacy. He's got an exceptional portfolio of proprietary technologies, patents, and a wealth of data in data vaults, which have been scientifically validated by third parties. And so he stands at the forefront of innovation in tackling this devastating group of diseases. While today's presentation is on the brain story, he has a portfolio of other pipelines of diseases using the same model. Now, why I think this is a solid, compelling investment opportunity is that it stands unusually on a solid bedrock of 5 pillars of science. The first of these is that Rene has patented several versions of this human brain model. He's far ahead of the competition. So this is the most advanced US patented human brain models available. They're not simulations. They're actual living laboratory tissue taken from a patient with a neural disease and then grown in the laboratory. So this is precision medicine in the test tube, in the dish, which can then be interrogated and experimented with. Now, this is in line with the latest thinking of the FDA, who recommend this type of model because it doesn't use animals. And they've got a Modernization Act from 2023, which encourages researchers to use this humane model, which Rene has been a trailblazer in over the last 10 years. The second big item.
Can I interrupt you 1 second? Because
Sure.
what it really is fascinating to me, and perhaps others, that taking tissue and asking the diseased tissue what it is so you can identify it and eliminate it seems to be something that one would have thought about many years ago and done.
Yes, you would think so, but I think it took a lot of stem cell development work. I mean, Rene can probably answer it better, why it took so long, but it's taken Rene 10 years to perfect it, because the ability to grow human tissue in the beginning with cell culture was very non-specific. Only certain types of cells grew. For example, cancer grows very well because the cells are immortal. But to take a non-specific cell like a skin cell, to switch off all the non-brain parts of the DNA of that material, and then to encourage only the brain part to grow, so that you end up with what is effectively an embryonic mirror image of that patient's brain, is quite extraordinary.
Yeah.
Quite extraordinary.
Amazing.
And I think it's the most important question that a lot of people don't get, that this, and Rene will be able to go through it in detail, but this is the thunderclap idea that you've now got something which is specific to that patient, that you can ask which genes are switched on, and he's able to do this with hundreds of genes at a time, and which genes are dysfunctional. And then he's able to compare those genes with diseased tissue from other patients, including post-mortem brains. And then, even more remarkably, he can add a drug to the dish and switch off, if you like, the bad genes. And as he'll show you, take the red genes and turn them to green as they reverse under the drug.
Yeah, super
And
cool.
so the next stage, super cool. And the
Yeah.
next stage is to see, will this work in vivo, in large numbers of people in order to get the FDA to approve.
Got it.
So that's just that's a fabulous question, showing you really understand what we're offering. So Neurxstem's scientific prowess shines through its arsenal of patents for drugs. Some of these drugs are repurposed. In other words, they've already got the required safety and probable efficacy in humans. And by repurposing them, we save a tremendous amount of time, because it could take a decade of research. So he's got both the model and the drugs, and he seems to be able, at a genomic level anyway, and a metabolic level, to reverse the tide of aging and light up the pathway towards either holding these diseases or even rejuvenating the aged brain. Now, this is backed up with clinical biomarkers. These are, if you like, beacons of success, which are genetic and correlate very well with clinical markers. And so the whole story is becoming very coherent. The third strong point is this jewel box of biomarkers, some of the hidden secrets of Alzheimer's and aging, which apparently can be detected even at birth, are available because Rene has been studying them for all these years, and he's found that which genes are relevant and which seem to light up, and then he compares them with the literature and with other patients. And using this, he's able to study the age-related changes at the earliest stages of life. And from this knowledge, we might be able to intervene in healthy aging as well as in well patients, but also specifically develop a new era of proactive precision and preventive care, where we can reshape the trajectory of people whose lives were on the pathway towards early cognitive decline, Alzheimer's, or worse. The fourth element is the data, the soft bank. In this is a treasure trove of data, which is very rigorously scientifically done. He'll show you some of the validations and correlations. And it's a repository of scientific knowledge, so data mining of what has been stored over the decade is another source of potential commercial value as well as human and social value in unleashing the knowledge and possibly wisdom that lies there. So this enables us to pinpoint potential targets for therapy, and the drugs can therefore be safely tested on that specific patient's actual tissue before you apply it to the living human being. These data-driven insights then pave the way towards other breakthrough treatments, which we hope can transform the outcome for the millions of patients around the world that have these diseases. The final important point is that the data is all scientifically validated. It's been corroborated by human clinical data, post-mortem and clinical, and also by the literature. And we now have these proprietary drug targets. So both the model and the drug and the mechanism is understood. And so what's happened is that so exceptional is the work of Rene and his team that he's attracted very competitive financial support from large government bodies like NIH and the Department of Defense. And these highly competitive grants testify to the validity and the confidence and respect that these grant-giving bodies have for Rene's work, which up to now has been funded entirely through Rene himself and the grant-giving bodies in a non-dilutional way. Now, if we put all this together, by investing in Neurxstem, you could become an integral part of this exceptional journey, where rigorous scientific validation and visionary funding combine to converge and so shape the future of Alzheimer's and brain disease investment. So we're really inviting you to join a scientific drug discovery movement. It's not just a financial investment. It's a whole new field of medicine, which Rene has unlocked. And with a robust intellectual property portfolio, pioneering drug translation and patents, we are about to move from the brain lab to the actual brains of patients with these conditions, and hopefully turn the tide on premature brain aging, cognitive decline, and ultimately Alzheimer's disease. Thank you.
Thank you, Peter, for that great summary. What I'm going to do now is to give you a little bit of my background. I have a background in chemistry and chemical physics, in genomics, and also in neuroscience. We have been funded, as Peter said, by the Department of Defense and also by the NIH. And this year, our work has been selected at 4 different business pitches as top innovator in Berkeley, Silicon Valley, New Jersey, and soon I'll be presenting a lot of our work at the Tech Connect World Conference in DC, all of which speaks to the excitement that all of these conference organizers have about our work. Where are we located? We are located actually in Heath, Ohio, and you may or may not know much about this place, but it is a phenomenal high-tech place. It's got Microsoft, Google, SpaceX, Boeing, Air Force, and we are actually the David in these giant Goliaths. We are in a phenomenal place, high-tech location. It might be the next Silicon Valley because Intel's come there too. The ask, as I said before, is for an addressable market of over $250 billion. Across 6 years, we are looking at $150 million in tranches of 10, 27, 56, and 57 million. They are to bring the drug through toxicology, phase 1, phase 2, and phase 3 for 2 novel drugs. What are the money? What are we going to spend the money on? Preclinical work in the first 18 months. Then we are going to go into phase 1, phase 2, and phase 3 for a total of 150 across 6 years. So that's just upfront what we need to get to an inflection point in the market. So what's the problem you're trying to tackle? There are no effective drugs or early diagnostics for any of the brain disorders or diseases that I'm going to cover. The biggest challenge was the assumption that rodents were good models for human brain disorders and diseases. And as it turns out, it has disastrous consequences for patients because to this date, we hardly have any drugs that work. So back in 2015, we predicted the need for a reproducible, robust, and relevant model for humans. We filed our first patent back in 2016 and made human brain models. 2023, the FDA has, through its Modernization Act, requiring no animal testing and a diversity plan. For this, there is no other option but to use human models, and we have been almost a decade ahead of the FDA in our prediction and why we engaged in such high risk at the time work. So what do? We take skin cells from patients through a proprietary process. We make brain mimics. They have all the major regions of the brain and all the cell types, and they are 98% identical to the human brain, which makes us very unique, which is why we have a patent on it. How do we do the science? We look at these brain mimics from a normal donor, and we compare that to somebody with a disease, and we look at all 20,800 genes expressed in human chromosomes, and we look for the differences in expression, and they are shown over here in the yellow circles when we're looking at, say, 4 genes. When we do that, we are now able to go through this process, and we're able to get the expression of all the genes that are different between normal and a disease model. We're also able to look at the metabolites, which is on the left, shown here on the left. And from that, we are actually able to derive the milestones that we're interested in, the early diagnostic markers, the drug targets, and then the drug discovery process itself. I'm going to walk you through just one example of Alzheimer's disease initially. So I'm just showing you some data because we want to be, for those who have a science background, you begin to see the rigor in the work we've done. This is following the development of the model through time at different times. D0 is day 0, day 1, day 3, all the way to week 12. And on the two axes are individual brain mimics. And because you see straight lines, these genes are all highly reproducibly expressed. We have done this.
Rene?
Yes.
May I, for 1 second?
Yes.
Is the drug that you introduce to fix the problem, I'm sorry to be so pedestrian about it.
No, go ahead.
Is also part of your portfolio, or is it a drug that wouldn't have otherwise been used for this, but you happen to know that it works?
So are you asking me, is this a repurposable drug or an entity that already exists? Or are you asking, did we make a new entity? Is that the question?
Did you make a new drug?
No. That's the beauty of the speed at which we can go, is the recognition of the target, which I will get into, and then the use of an existing compound that makes it so much easier to get to the endpoint, which is why it won't take the amount of time and money that it would take normally to create a new entity. So we have filed patents on the use of this drug for this, and has not been recognized as being useful in Alzheimer's.
Yeah, that's what I was looking for. Thank you for that.
Yes, thank you. Yeah, very nice point. So here you're looking at the top is familial, from a familial patient, Alzheimer's, and the bottom is sporadic. Again, I'm just trying to show you the rigor in the science, because without rigor, you can't actually get any marketable product. And so again, we look at the differences, like I said before, and then we look our data output, which is filed in patents all the way back from 2016 to 19, has been corroborated last year independently by AD Genetics. Here's some 72 genes that they say is correlated with Alzheimer's. And out of them, everything that's in a green box, we already knew were correlated with Alzheimer's through our model system, which gave us the confidence that what we're doing is relevant to humans and is not necessarily something coming out of a mouse. Our data is also corroborated very heavily by AD postmortem data, where they looked at the differences between those brains that are Alzheimer's versus normal. And here is a summary of that. The brown circles show you the 2,100 genes that are in postmortem AD brains. And in the blue circles, what I have is from 4 different brain mimic models. 2 of them are familial, 1 is sporadic, and 1 is ApoE, which is the largest number of patients that carry ApoE. We then look at what's common between them. There are about 500 genes that are common. And then from there, we are able to do a deconvolution of very large data sets using a mathematical method. And we are able to identify early diagnostic biomarkers at birth, as well as drug targets. So now I'm going to get into the 80 therapeutics of the drugs we identified that Arthur was talking about, and the efficacy profile. But I'll stop here and see if you have any questions.
So one of the things that seems to make sense is that if you're able to identify early on the biomarkers that would suggest that Alzheimer's is likely to happen, is the idea that you would just treat that in advance to thwart it happening, or keep it from happening sooner?
A really good question, Arthur. I will get to this towards the end of my talk. But however, let me just premise it by saying there are 2 strategies that we are going to unleash. One of which is, if you currently have Alzheimer's or in the early stages, we have drugs that we believe can be very efficacious in treating the disease, especially at the mild cognitive impairment stage. The clinical trials will determine how well they do at the different stages of disease. However, we have also found that there are environmental factors that are accelerating the susceptibility at birth. as well as nutritional factors that are exhilarating.
Makes so
That's
much
the
sense.
beauty of what we have found. And so in your early stages, if you have an identified risk, you can alter that risk to the point where you may not even get Alzheimer's. So this is for multi-generational changes.
Yeah.
And I'll go through that in a second.
Yeah. One other thing, just so
Yes.
I don't forget to ask it, at some point, it would be super interesting to understand why the NIH and others decided to deploy non-dilutive capital into what you're doing, just from a, perspective of, okay, why did they fund you? And so at some point, it would be really interesting to know that.
Yes, I will get into that. I want to say that the NIH has funded us for an entirely different portfolio disease, the opioid crisis. We have modeled the opioid crisis in the dish. We have a pending grant, our second round of funding that has done extremely well, where we now have drugs that are likely to block the effect of fentanyl. It's a phenomenal story that I'll be telling, and I don't have time to do it today.
Yeah.
I could get into that at the bottom if we have time, where we are dealing with a crisis in the nation, and we have ways to do it. Similarly, the Department of Defense funded us because we are addressing a crisis in PTSD. So those are not part of my talk today. I'm going to focus on something that we believe is at an inflection point for our investors. The other ones will be at an inflection point, be perhaps thinking about a year and a half.
Good. Fair enough. Thank you for that. Yeah.
Okay. So now what I'm going to tell you is about the phenomenal discovery of compounds that are very efficacious in reversing the biomarkers of Alzheimer's. So what I'm going to show you is in red, a column in red, which is the difference between Alzheimer's and normal of hundreds and hundreds of genes. And in green, what happens to those genes when we apply a drug. So on the left-hand column are all the biomarkers. We only picked 15 of them because they're clinically relevant. And we're looking at sporadic Alzheimer's plus and minus the drug. And what you can see in green is not only are the genes, those that are downregulated, are upregulated, and they reverse throughout all of these genes, either in the magnitude or in the polarity. And when we do this across 4 different models, we see over and over and over that those biomarkers are present and are reversible. The ApoE4 shows the greatest number of genes correlated with Alzheimer's. And there's a reason why this is the most penetrant form of Alzheimer's today. On the right-hand side are the clinical symptomologies, and I'll show this blown up again. And what you're looking now at is a completely different way to begin to assess Alzheimer's throughout your life by looking at what we call proxy markers, blood pressure, vision, sleep dysregulation, thyroid hormone dysregulation, and are all in yellow. And currently, the current standard is memory and cognitive only. To do something at the MCI stage, you will actually need all of the other ones, or even earlier, to be able to see how the drug actually works in a clinical trial. So this is basically what they do today, and we are so far ahead in biomarker-driven changes that the clinician can use in the clinical settings when they do the trials. And so the future is personalized therapeutics. It accounts for diversity. We now see diversity among men and women, and also between different ethnic groups. So all of these can be addressed only by a human model. Currently, there are no human models in play in clinical trials. They're all mouse or some form of rodents or other species that are essentially irrelevant to human health, as far as I'm concerned. So our trajectory is preclinical, about 1.5 years, where we finish the safety and toxicology, very likely to work. And in some cases, we may even be able to skip this and go straight into phase 2, depending on the drug. We have 2 drugs. The bottom panel is 2 drugs here, a single site trial, a multi-site trial, and then a phase 3 that we anticipate across 6 years. And what is the current result for Alzheimer's clinical trial? This is a drug from Eli Lilly. And you can see there's plenty of room for something better than what is out there. 31% of the patients had brain bleeds, which is, in my view, really not an ideal way to bring a drug to market for Alzheimer's disease. So we are just saying that our work very favorably competes in an area where there isn't anything that is safe and very efficacious. Our bigger portfolio is very large, as an Arthur got into it a little earlier. I only talked about the Alzheimer's side of it. However, we have a very large database of all the diseases. The models are all made. The patents have been filed. I'm only going to talk about aging. Very fascinating story that is playing out. So we looked at a disease called progeria. Why did we look at it? It's an early aging disease. And for some of you, you might recognize this movie, Benjamin Button, where it was a science fiction kind of movie where somebody was born with progeria and then they reversed the whole process. It was a movie, but we asked the question, can you actually do it? And here is a little bit about longevity and aging that we know about in human populations, the blue zone aging, and these are the 5 places where they're known to occur, and then progeria on one end, and blue zone aging, where they age very healthily. So the question is, if nature makes this difference, then what makes them so healthy? Can we ask that question? And we did. So we got an early jump start when we looked at our 4 models and we discovered that 2 genes connected, linked to progeria, were actually present in Alzheimer's patients in our models, which told us that Alzheimer's, at least in part, is an accelerated aging disease. We're the first ones to show that. And there are our patents now. So we then asked, what are the hallmarks of aging? Remember, this is not quite a disease other than Werner syndrome. So there are a number of criteria by which aging is benchmarked. And what you're seeing here is in the first column, along each one of these, it's too small to see, so just follow the red columns, is the difference between progeria and blue zone, normal people. The second one is Alzheimer's and a healthy aging individual. And the third one, which is the most fascinating of all, is what does the drug do to all these biomarkers? And by just putting it in green, you can look at the numbers later on, you can see it virtually reverses all of most of the biomarkers of aging that is currently that are considered of relevance. So what I'm trying to say here is that it is becoming clear that we may be able to move the needle of aging as a whole in a direction where everybody's epigenetic clock, a biological clock, is synchronized with a chronological clock, which is your birth certificate. And that we are, every one of us is somewhere between progeria and blue zone, and there may be a means to move us all in a much healthier way towards healthy aging. It has major implications for the economy, for your health, for your ability to work, and not really be disabled by crippling things that occur as you get older. So that's a second portfolio that we're moving forward. We have all our patents that we talked about. We have one issued already in US. It's pending in China and Europe. So this is the intellectual property side of it. The capitalization I mentioned. Arthur asked about the NIH funding. We have one which is on opioid, a second one on opioid addiction that is in council, that is pending. We have opportunities to present all the stuff. So up to like $11 million to de-risk these projects, at which time they become an important part of getting investors on board to move them to the markets. I would say not quite yet for all of the other ones.
So could you jump back to that prior slide for a second? Just
this one?
Yeah. No, the other one, yeah. Prior to this. I'm sorry, one more. So for those of us that are not scientists, right?
Yeah.
This third column is using a drug plus
Alzheimer's.
And yeah. Right. So what is it that's different that you're doing by not just using the drug alone.
If you don't use the drug, this is what you see here,
Yeah.
the red column.
Yeah.
And that's the difference. That is the difference between an old person that is aging normally versus Alzheimer's. And there are a number of Alzheimer's models here. All 4 models are included in this particular column.
Yeah.
And this is progeria. We made the progerial model from a donor, from a patient donor who had progeria from the skin cell. We made the model, did exactly what we did with Alzheimer's, and we asked, are they correlated? And they are beautifully correlated. So we now know, we predicted based on 2 genes that aging was a factor. But when you looked at progeria, shockingly, the exact same genes are dysregulated. So then we said, if that's true, that is an accelerated aging model, could you reverse that process? And here, you could call it Methuselah science, Benjamin Button science, whatever you want to call it.
Yeah.
I mean, that's the common lingo. But the bottom line is aging is modulatable. And here's the evidence that it is. And we're very excited because this is not a disease-specific thing. What this is a condition that disables everybody globally. So the market is huge. And we believe that with the proper clinical trials, we'll be able to demonstrate that the drugs, and we have multiple drugs, by the way, I'm only showing you one, that can actually enable you to live a healthier life. And there are other issues that it may not be just brain specific. We believe these drugs, because there's commonality between brains and other organs, may actually reduce osteoarthritis and a number of other conditions that disable us, including increased proclivity for getting cancer. Everything that happens in progeria is accelerated aging. And we know that they have cardiac disease, cancer, osteoarthritis, and the gentleman that I showed you in the slide before, he is Mr. Adams. He died at age 17 from the complications of this. It's a very well story that was covered in CNN.
So the difference between taking the drug by itself and doing it the way you suggest here is that you have more specific guideposts because you've identified certain markers that you know the drug will work well. Is that right?
Yeah. So what we are doing is we are at a nexus in aging and Alzheimer's. We have identified targets that we're not telling you what they are currently. We're protecting it primarily for investors. The investors will know what the targets and the drugs are, but we have a right to protect that investment to the investors. So at this point, we're not making, So it's a matter of simply a legal IP thing that I'm not disclosing that yet.
Fair enough.
But it will be disclosed. It'll be disclosed to the investors. And so the idea is there are at least 2 different targets, Arthur. We don't look at individual genes. We look at networks. And I didn't want to get into that because it becomes very mathematical and much more like you would look at something through like an electrical engineering, which is why on our team we have mathematicians, electrical engineers. I'm a chemical physicist. We look and do things differently from clinicians and biologists and neurologists because we understand how a system works. It's about complex system biology. How does it actually work? And once we are able to find out where exactly things are starting to go wrong early in the process, if I may, I'll give you a metaphor. The Mississippi floods every year, right? And our typical strategy is to put sandbags along the cities and keep them from flooding, and the sludge comes down. I used to live in Louisiana, down all the way down. What we did is we went all the way upstream and said, where are the polar ice cap melts and where's the rain and what actually causes the flooding? And then you know what we did? Put a dam.
Totally
as
makes
soon as
sense.
we put the dam, everything downstream from there
Changes,
itself.
yeah.
That's what you're seeing here.
Yeah.
It's a metaphorical way of saying that these are all correlated. These are not individual genes functioning on their own. They're actually parts of different networks. And by finding out where do you want to put the dam, you all of a sudden flipped the switch. It's like, that's why we use the red, green, because looking at numbers and large number of genes is not aesthetically pleasing for a general presentation.
Yeah, but you illustrate it by the green just across the board.
The simple way to illustrate, you go from red, which we all understand is, it's blocked, and green, it flows. And so when it starts to flow to normalcy, you are going to restore block functions that are a domino effect all the way down throughout your life and aging, and in Alzheimer's, possibly in many other diseases that we're looking into now. Maybe one day it'll be cancer.
Yeah, very
I'm
good.
just throwing that out wildly because we think now that this model is working and our strategy is working, we will take on cancer one day, but not right now. That's not on the table. We want to be sure that when investors come on, that we are successful. And at this point, it's aging and Alzheimer's.
So there's a couple more questions, but continue so you can get to what you need to get to.
Okay, so these are the patents. This is the funding that we have, and some of them are being submitted. This is once again just a reiteration of what we're looking for. We think we'll be very successful. We have 2 drugs because that gives us a greater safety margin of one of them or the other doesn't work. What Arthur was asking, and I wanted to address it here, genes are not your destiny. They only increase susceptibility, but what actually then starts to play out is the environment and nutrition. By detecting things at birth, we'll be able to intercede somewhere between infancy, adolescence, so that you don't actually go down. This is very general, not necessarily just Alzheimer's focus, but it does include Alzheimer's. We can detect Alzheimer's at birth. And then we're changing the way we do business. All of neurology was reactive. We'll be doing everything predictive at birth for all brain disorders. And then I live in Ohio, and we hope to see what the Wright brothers did back when they started aviation. Today, we are looking at what the future looks like, patrolling the skies. We were considered pioneers back in 2015 when I made the models. This is CNN says, these mini brains will help solve cancer, autism, Alzheimer's. We do hope that it'll do that. We're looking for blue sky cures. I hope I've demonstrated to you that at least for Alzheimer's and aging, we have reached that inflection point. We are trying to move with many of the other ones. And then here are our team. We have Clay Marsh, who's involved in personalized medicine. He's an advisor and a referee. Peter, you met. Dean is our CFO. Joseph Armstrong is a consultant. He worked for Merck, and he'll be very helpful in our drug development strategy. And then a number of others that are referees for us. This is my team. Susan McKay was one of the co-inventors. We have business advisors, Johnny and Manoj. And then our legal team, Marshall Eisenberg in Chicago, and our intellectual property lawyers. And this is the organization of our thing: clinical, R&D, financial, legal, and IP. I won't get into all the details. I showed you some of the faces. And then finally, a big thank you, and I'm happy to take questions. I think we have about 15 minutes, so I'll stop there.
We're good. So what are the things that you found so far that are possible pushbacks? Meaning, is pharma on your side? Are there people that would not like you to succeed in a commercial way that could stop this from happening? I don't mean to be negative, but there's powers that can.
Yes, Arthur. We have talked to all pharma, undoubtedly. We have one of them that is interested in our model system to develop their own drugs. Okay, there is a conversation ongoing. It's out of Japan. The challenge with pharma is, as you know, they are like an aircraft carrier. Once they are heading in a direction, whatever it might be, they're not about to turn around. They have invested billions. So it is in their interest to reach the markets as soon as they can. And we've seen for Alzheimer's, several of them that have been big flops. Biogens, they got FDA approval. The markets rejected them. The efficacy was very poor. Nobody wanted it. Lilly has got its drug now. It's not yet FDA approved, but there's a big red flag on that 31% of patients that have brain bleeds. And I think that I don't want to say, it has some efficacy, but we can do a lot better, Arthur, a lot better.
Yeah, 31% sounds bad, right?
Yes, it's bad. It's 1 in 3. You're playing Russian roulette.
Yeah, right.
So my thinking is I don't want to get into what pharma does or doesn't do. I think we want to offer a much better alternative for all of us, humanity as a whole. And we are showing you, evidence that we are on the track, we have the right model, it's corroborated, it's doing well. So I'll let Peter have the last word on trying to answer your question, if I may.
Sure.
Well, very quickly, there are obviously a lot of pushbacks, which, I mean, being scientists, we expect that, and actually it makes the science better. One of the pushbacks has been people didn't believe Rene's model when it first came out because it's better. I won't mention the name, but there's a famous lady researcher in Cambridge who's also doing similar brain neural organoid work, but her brain model doesn't have the complexity that Rene's does, and also doesn't contain all the cell types. And she hasn't been able to do, and I think it's really got to do with the fact that Rene, fortunately, is not a doctor. He thinks in an engineering kind of way, a physics kind of way, electrical way. And what he's done is he's identified the control networks that seem to be involved in Alzheimer's disease, and it seems also in progeria, and probably normal aging. And by knowing the network, using the Mississippi analogy, he kind of knows which drugs are going to be effective. If you understand the whole control system and which goes up-regulated, which is down, and so on. So it's a very sophisticated way and quite different from conventional science. And that's what I think makes it exciting. I'm a bit of a student of medical history. And, we're sitting, I think, in historical terms at a Salk moment with polio, or a Jenner moment with smallpox, or Semmelweis with hand washing, where if people tell Rene he's nuts, we know he's on the right path.
Right.
Because that's the history of medicine.
Yeah.
Because he threatens the establishment in a benign way, but actually, they may all be barking up the wrong tree. And Rene seems to have cracked it through novel thinking and researching and good science.
Yeah, I'm going to say out loud, there are people that I know well that are focused on longevity process, right? Because once you have more money than you could ever spend, and you're reasonably healthy, and your family's safe, and the world is a disappointing place, but it's an equally impressive place because people are clever and they innovate, like in your case. But once you, at some point, there's many people that are saying, okay, so can I live in a healthy way a little longer, right? So this is not a new thing, but there's a sentiment underway that this can be done, and we're at the beginning of being able to figure out how to live longer. I mean, there's people that are on the extreme of that and say, we'll live forever, but that's a whole other thing. Would you say that aside from the lifestyle issues of identifying, let's just talk about Alzheimer's, and the expense associated with treating that, just the family expense, that the longevity is part of the equation here?
It seems
yes,
to be.
Go ahead, Peter.
Sorry, Rene. Rene had a brilliant insight in looking at the progeria patient. So again, to repeat, this is looking at the skin cells of this unfortunate person who died young, as they tend to. It's all in the public domain. And by making the link, and I as a pathologist, we always knew that Alzheimer increases with age. In fact, I can tell you it overlaps also in Down syndrome. People who develop Down syndrome, some of them end up dying quite young in their 30s and 40s, and their brain changes are identical to Alzheimer. So we've got three different models. There's Alzheimer itself, the progeria model, which is accelerated aging, and then there's the Down syndrome model. But just to add two other things, in addition, and being an older man myself, I'm quite interested in this stuff because there may be things we can do or avoid which will enable us to enjoy life. But here's the thing. With Rene's model, we can open it up, and those companies interested in aging, instead of reinventing the wheel, could, if you like, buy time on Rene's land and experiment using this superior model. Likewise, pharma companies who are chasing other drug solutions can do the same. And so the technology could be sold as a kind of pharma platform.
Like a licensing deal, yeah.
Yes, that's right. In addition to us doing our own discoveries in science. And just to give you a little clue, my special interest is in cancer. And one of the main criteria, a hallmark of cancer, is immortality. So you can guess that if we start studying cancer genes,
cancer may give us the secret, right?
Using the analogy with progeria, it's very likely we'll find, or Rene will find, the regulatory networks that are involved. And then we can start to, we don't have complete understanding, only enough understanding as Salk did, and Jenner did, and Semmelweis did. Okay. Well before these agents were discovered, they said, let's interrupt it here. And that's what Rene's doing.
Yeah.
Enough understanding to change the course of history.
Totally makes sense. Now we have another question about the data. The data that you have compiled, does it rival others? Is it bigger than others? And it's this is my editorial for the question is, those who claim to have an idea of what the future brings, in basically say whoever has the data is going to win. So how's your data compare? Because you seem to have a lot to others that might have the repositories of data like that.
Right. So let me walk you through this systematically. We made the model. We looked at all the genes that are expressed. We became convinced it completely replicates the human brain. We then did that through Alzheimer's, autism, and a number of other diseases where there's a genetic susceptibility. And it became very apparent that when we looked at our dysregulated genes compared to normal, they were actually correlating with Alzheimer's data, both genetics and postmortem. What I want to tell you is, how do you know our data was correct? Because we made it public in our patents years before the rest of the world caught up. Okay, the postmortem data and genetic data was published in 2022. My patent they filed in 2016, 18. So that's how you know that we didn't make up the data. The data was already public in our patents. Now, having said that, not all the data is public. Some of the data is being protected because we are looking at how that gives us insight into drugs and therapeutics and diagnostics, which is in the interest of those who are investors to protect their ability to bring it to market and protect it all the way through. So that's number two. The database is huge. All the models I've made there have been done, including opioids, PTSD, brain cancer, on and on and on. It's a huge database, and we are deconvolving them using the exact same strategy. The idea of Alzheimer's is used as a prototype. Can we go all the way? Can we show we can identify the targets? Can we make a drug or identify a drug that can reverse the process? It was an internal challenge to show that our strategy is phenomenal, that it is a revolution in how medicine should be done. And now we are looking at the final proof. Let's see it in patients. And we believe that our model is so well correlated with Alzheimer's that the odds are very, very much in our favor that the trials will go through very, very well. Does that answer your question, Arthur? Or
so I'm kind of a data junkie in that in my businesses, I've bifurcated companies and taken the data and put it somewhere else and monetized it. So I think that the not because I was smart, it's just that I paid attention to others who were smart, right? And so I think that data wins, right?
It is correct. I come from the world of physics, and it's always chemistry and physics initially. It's about the data and the metrics of reproducibility, robustness, the R-squared coefficients on the data. How do you know it's accurate? How do you know? Well, you can get a lot of precision in a mouse data or anything else, but is it relevant? And that means it has construct validity. That's where when we compare what's out there in the postmortem, what we have filed a patent, and you see these hundreds and hundreds of genes with a p-value that is minus 300 or something like that are correlated. That is not a stochastic correlation.
Yeah.
That's what gives us the confidence. And then when you reverse those, the drug, become confident, oh my goodness, this is the real deal. We can change people's lives. Coming to aging, you asked how does that thing compare? We have looked at some of the other drugs that people claim help with aging, rapamycin, metformin. And the answer is yes, they marginally change the progerial trajectory, but none come close to what we have done.
Yeah.
We know that already. So we are so much better. There are many ways to skin this aging cat.
Arthur, if I may very quickly, sorry, just to your point, we could monetize this company by selling the data alone or allowing others access to mine the data using the model. Because if you think about the numbers, I mean, Rene would have figures, 20,000 genes per person, per patient. And then he's got many studies, and he's done this over a number of years. So if a pharma company wants to come and, you know, pay to have a look or buy the data or license it, or the other thing is, we can exit by selling ourselves as a data company.
Yeah.
So not only are we a drug discovery company, but we're also a massive data warehouse, which could be monetized.
Yeah, totally makes sense. Well, this has been super helpful. We do have to run, but I want to make sure, if it's okay with you guys, can we do this again?
Sure.
And I'm not trying to retell your story, but I think that the longevity headline is really important to get people to understand what you're doing. Not a bait and switch, but just that is sort of the hot button of the day. Aside from the Saudis buying global golf. So yeah. So super cool. Thanks so much, Peter. Nice to meet you.
Thanks so much. Lovely to meet you.
Good to see you again.
Arthur, thanks so much. We'd love to be on again. Love to be, increase the focus on aging. We really do believe that quite by accident, through the Alzheimer's portfolio, we discovered a new novel way to address aging. And it's a matter of doing the clinical trials in various model systems to establish that this works in a full human being and make history. We don't have to suffer the way we have and has made the assumptions that aging was just natural. It's not. And there are ways to enable that through diet, nutrition, watching the environment, and enabling some of that with drugs. So I think they're all going to play in together.
Sure sounds like it. Super.
Yeah.
Well, thanks again.
Thank you, Arthur, so much.
You're welcome.
Pleasure being on this.
Thank you. Great fun. Thanks.
Thank you.
And as I always say, thank you for spending with us the only thing you can't make more of, and that's your time. Although you guys make
Very well put. Thank you.
Thank you.
Delightful.
Okay, guys.
Thanks.
Thank you, Peter. Bye-bye now.
Thanks, Rene. Bye now.